Vinit Mahajan, my co-moderator, and the next speaker is a vitreoretinal surgeon scientist at Stanford, and he directs the molecular surgery program at Stanford, and also cares for patients with genetic retinal diseases. So Vinit, you probably know a lot more than me about these conditions. And also his team is developing gene and small molecule therapies, which is very interesting. So I'll hand over to you, Vinit.
Yeah, thank you. And I think, Stefan, you began to touch on a few of the topics that I'll be briefly going over. I have no financial disclosures. A lot of our funding comes through the National Institutes of Health. And we're going to talk just a few minutes about the relationship between inherited retinal disease and optic disc drusen, and how human eye diseases can be modeled in mice. So I'm going to show just a few clinical cases that I think we were touching on briefly. So here's an example. It's a 19-year-old male, 10-year history of night vision issues, progressively worsening vision, no family history of eye disorders, vision in the 20/50/20/60 range. And I'll show you the back of the eye in just a moment. But an ERG that has an extinguished rod response, but a relatively intact cone response. When we look into the back of the eyes, we can see there is peripheral pigmentary degeneration, even some pigmentary changes within the macula. And on autofluorescence, you can see all the dark spots indicating RPE atrophy. But what's really nice here is you can see these bright white spots popping out of the optic nerve. And those, of course, are the optic disc drusen. Earlier this morning, there were some nice OCT images showing the disc drusen at the optic nerve. And in one of the previous questions, we don't have histology of many patients with disc drusen, but perhaps the OCTs will give us an indication whether disc drusen in genetic retinal diseases is similar to the sporadic cases. These are some additional images. This patient underwent genetic testing, and we were not able to identify any genetic cause. There was a variant of unknown significance, but no clear etiology. This is a second patient, a 33-year-old woman, 4-year history of progressive night vision, a very significant family history of retinitis pigmentosa. She still has good central visual acuity, also abnormal ERGs. And I'll show you the back of her eyes. We can see typical pigmentary degeneration in the periphery, some changes within the macula. This is her right eye. This is a color fundus image of the left eye, very similar. The autofluorescence image again shows RPE degeneration in the peripheral retina. You can see a hyperfluorescent ring within the macula, a finding in many RP conditions. And then a few of these bright white spots again at the optic nerve head. The OCT here in the macula shows thinning of the outer retinal layers. You can see from this pedigree, she has quite a significant family history, and she was found to have a mutation in the rhodopsin gene. Here's our third clinical case, and this may sound a little familiar based on our last talk. This is a 19-year-old male with eye abnormalities since age 6. No family history. His visual acuity is now 20/70 and 20/80. And what's interesting is that he has high hyperopia in both eyes. And what has happened, these are serial OCT images, is that he's developed cystoid macular edema right near the fovea. Fundus images show some pigmentary degeneration and a few yellow spots, tiny yellow spots in the periphery. If we, in this case, we got a B scan, and you can see a hyper-reflective spot indicating the optic disc drusen, and the axial length of the eye is quite short. The patient underwent genetic testing and had mutations in the MFRP, or membrane type frizzled related protein. And as Stefan mentioned, the actual diagnosis or name for this condition is nanophthalmos RP foveoschisis and optic disc drusen. And so by definition, these patients have optic disc drusen. We know MFRP is important for eye development. It has some importance in phagocytosis. And I'm going to transition now to the second part. The MFRP is actually, the gene has been known for quite some time in the field of mouse genetics. And many of the retinitis pigmentosa diseases can be modeled in mice. And so I'm going to briefly just show a few figures from one of our papers that used gene therapy to treat a mouse model of this disease, in particular correcting the axial eye length. And so here, panel A shows the normal axial eye length, panel B, the shortened axial eye length that we have. And then one of our patients in panel C that was phenotyped and found to have an MFRP mutation. The mutation in this case was near the C terminus of the protein, but as I've shown in the bottom panel here, you can have mutations, loss of function mutations typically, that span the entire length of the MFRP protein. We don't know a lot about its function, but the RD6 mouse has been around for a very long time. And in panel A, the mouse will show tiny hyperfluorescent bright white dots throughout the fundus. And you can also do OCT in mice. And in the right half of each of the panels, we're showing after we do gene therapy to replace the normal MFRP mouse, we can diminish the number of those abnormal white spots, and we can actually increase the health and size of the retina on OCT. We can also perform ERGs on these mice, and gene therapy will rescue the ERGs. And then if you can get good pupil optic nerve sections of your tiny mouse eyes, you can actually very carefully measure the axial eye length. And here we have restored the eye length of the mouse. One of the things we really like to do in our lab and research is identify proteins that may indicate pathways, molecular pathways of why we're getting certain kinds of eye disease. And so MFRP is expressed in the RPE, and what we're showing here is a number of the proteins that change over time. There's a heat map on the left, and in the green panel, you can see proteins that stay the same. In this column, these are the control mice. In the mutant mice, we can see a group of proteins that increase, and after gene therapy, these increased expression of proteins is restored to normal. And we can go through and look at a variety of proteins that change in the MFRP mouse and those which are restored after gene therapy. We can then begin to try to correlate these molecules with different phenotypes. The focus of this paper was axial length, and we can find different proteins that are associated with scleral thickening, retinal degeneration, CME, and so on. And these then perhaps give us an insight into what are the downstream molecules that are actually triggering the various clinical pathologies that we see. Now, since Joyce has gotten me excited about optic disc drusen, we considered and we began looking at the database to try and identify proteins that might be involved in calcium metabolism and calcium regulation. The calcium phosphates are thought to be a principal component of the drusen. And in reanalyzing the data, probably one of the most important proteins that we found is this S100A9. These are antimicrobial or inflammatory proteins, but S100A9 is downregulated in this MFRP mouse. 100A9 is calcium sequestration. So perhaps this is one of the molecular molecules that may drive the formation of disc drusen. Now, one of the important questions is, do you actually see disc drusen in these mice? And I've shown a couple papers, including one of my colleagues, Steve Sang. In the autofluorescent imaging, you can see the bright white spots throughout the retina. You don't really see any these bright white spots in the optic nerves of the mice. So even though these mice don't fully mimic the human condition, they don't show the optic disc drusen, it's likely that they may be associated with the molecules and the pathways that, at least in humans, drive the formation of disc drusen. We have a good sense that inherited retinal conditions lead to disc drusen. Mouse models have been incredibly powerful at studying genetic diseases. We don't yet have a gene, but we can still use these models to identify pathways. One of the studies that we were involved in was actually a large-scale mouse knockout phenotype study, where several thousand genes have been studied, and we've been looking at the mouse eyes for phenotypes. Again, in many different genetic models, we haven't identified actual disc drusen, but there's an opportunity to look at calcium deposits, which may not result in the human disc drusen. So I've got a couple simple, straightforward quiz questions I'll just run through. And can optic disc drusen be inherited? I think we have a good sense that the answer to that is yes. Optic disc drusen cannot be modeled in mice. And I would say false. I don't think we've quite modeled it yet, but we can certainly identify or have an opportunity to look at pathways and some of the, perhaps the earlier processes like calcium deposition at the optic nerves in mice. So with that, I just wanted to thank everybody. Thanks for having me at the meeting here. And I'm certainly open to some questions or discussion. And happy to go with that. Thank you.
Let me see. Should I pose some questions?
Yes, please.
Okay, so there are some questions here. Are there any infants born with optic disc drusen, possibly just due to genetic causes, not environmental or other stress factors?
Yeah, I think that was also brought up in the last session. So I'm not aware of that. I think some of our pediatric retina colleagues who are doing EUAs very early may have the answer to that. For the LCA patients, I'm not aware that people have identified optic disc drusen at birth.
Okay, so there's a question here. Do PXE mice get ODD? And if so, are they a model for MFRP-related gene therapy to see if enhancing phagocytosis can help to clear the lesions?
Yeah, so I don't know the answer to that. We had looked at some PXE mice a while back, and I did not see optic disc drusen, although there was no consortium and large group to do that. What's interesting about the mice is that many of the mice we've been looking for a glaucoma model or any kind of optic nerve, and although in our large mouse screens, we can identify corneal disease, lens disease, retinal disease. ciliary body disease, it's been difficult to identify optic nerve issues. So there may be some resistance or different way that mice express a particular phenotype. Nevertheless, I think as we understand human optic dystrophism a little better, we can expand how we look for the phenotype in mice, for example. Somebody else may know about the PXE mice.
I have one question for you, Vinit. Did you do OCT on the mice, on the MFRP mice?
No, we had done OCT, but not of the optic nerve.
Okay.
So we were, again, focusing on rescuing the photoreceptor degeneration and then restoring the axial eye length. So we hadn't done OCT. What we're trying to do is identify the tissue blocks, and we're considering looking at different calcium stains just to see if calcium is accumulating. So sometimes you may not get as severe a phenotype in the mice, but we may be able to see some of the early things.
All right, great. Thank you very much, Vinit.